Right, good day Junior Tuckies, and welcome to this video in a series dealing with the DNA code for life. I am Mr. P, Mr. Prigger, who's going to be taking you through this section as well. Particularly in this video, we are doing practice question number 5 from the worksheet booklet that you should have in front of you now. So we are going to be moving straight into the question and doing our question analysis first. Okay, so the first thing we're going to be looking at is what is represented in the image that we're looking at. So the question luckily gives us a little bit of context. The diagram shows part of an mRNA molecule. So we can see the sequence of bases that are represented there. Obviously my nucleotide cannot really be identified here, or the different parts of my nucleotide. So this question is going to deal with the sequence of bases that we are looking at. Okay, and if we look very, very quickly at this, we can see that from left to right, we have a base sequence. It goes UAC, CGA, UAA. Keep in mind that this is an mRNA molecule, so we do not see any thymine bases in the mRNA molecule. We can see that there are 3 mRNA codons. So in other words, these sets of 3 bases form codons on the mRNA strand. And looking forward, we can see that if this mRNA strand was transcribed from DNA, there would have been a unique DNA sequence as well, sequence of bases, triplet codes that existed on the DNA strand that would have resulted in this mRNA strand. And if we try and reverse engineer this sequence, we can say that, right, U would have been transcribed from A from T, C from G, and so on. And we've got this whole DNA sequence that this mRNA formed from through the process of transcription. Now, as I said before, we do not have any thymine on our mRNA strand, and that is because every T that would have been there based on the DNA strand has now been replaced with uracil, U in this case. All right, let's have a look at the questions. So now, here's a little bit of a trick question. Question 5.1. How many anticodons are shown in the diagram? Now remember, this is an mRNA molecule. And mRNA base sequence result in every 3 of them forming a codon and not an anticodon. So in this case, I would go with 0. There are no anticodons present in this mRNA strand, only codons. Now, in the back of your mind, you should be asking yourself, well then, where would we find the anticodons? Okay, and we'll get to that. Right, so let's look at the next question, 5.2. Write down the complementary base sequence of the DNA strand that formed codon 1 in mRNA, or in the mRNA strand shown. Now, we're only looking at codon number 1. You can see that we've got 3 of them. We're only looking at the base sequence of the 3 bases of codon 1. And if we look at the mRNA strand, that is UAC. And if we're looking at the complementary base sequence of the DNA strand, we reverse engineer this, we go backwards and we look at the DNA strand, and we've got A, T, and G. Why have I put thymine in there? Because on a DNA nucleic acid, we will find thymine. Where we will not find thymine is in any of the RNA strands. And here we're typically talking about mRNA and tRNA. Question 5.3 says to us, explain the purpose of the specific sequence of codons on an mRNA strand. And explain, it's very important to remember that when you see this action word, this verb, we have to identify the what of the question and the why of the question. And in this case, if we're looking at explain the purpose of the specific sequence, so what's the sequence, what makes it unique, and then why is it unique, and what is it going to result in at the end of the day. So if we answer this question, we can see that each codon is a unique sequence of bases. That's the what. Now, what does this unique sequence of bases result in? So why the unique sequence of bases? Because each codon will then translate into a unique amino acid during protein synthesis. Now we've answered both parts of these questions. Question for 2 marks. Right. Now we've got question 5.4a. It says tRNA molecules carry complementary bases to the codons on an mRNA strand. Write down the complementary base sequence of the tRNA molecule. So we had a similar question before when we were looking at the complementary sequence on the DNA strand. Now we're going to the tRNA molecule. Now we're going to work it forward. We have A, U, and G. So U would pair with A on a tRNA, A would pair with U on the tRNA, and C with G on a tRNA. Why not T on the tRNA molecule? Because it is an RNA molecule, and we have uracil substituting for thymine. Last question over there, 5.4b. Describe the role of tRNA molecule in the process of translation, and tRNA has complementary anticodons to the mRNA strand, or the mRNA codons that exist on the mRNA strand. This carries the unique amino acid to be placed in a specific sequence during protein synthesis. So there is us describing why tRNA is important in the process of translation. And translation is the formation of the larger polypeptide or protein. Okay, so we need to make sure we talk about protein synthesis, because that's what translation is all about. Alright, I hope that this video has made sense. I sincerely hope that you've enjoyed it and it's created a better understanding for you. I look forward to seeing you in some of the future videos dealing with DNA Code for Life, and we'll definitely be seeing you again soon. Thank you very much, Junior Techies.